Okay, it was in the mid-teens, 2015 or so, that Richard LENSKI's lab, after the flowering of DNA sequencing technology, was able to go back and sequence the entire genomes of every stage of all 12 replicas of his bacterial culture. Well, in 2015, he and his team sequenced a total of 264 E. coli genomes, representing every replica, every few hundred generations, for 50,000 generations. And this is all the 17 highest mutations that helped the bacteria grow the best in his culture. So these are the best mutations. And just like those genes we looked at a few slides ago, if there are multiple mutations, multiple different mutations in the genes, that is a signature of their working by degrading or destroying the gene. He also looked at another 17 or so of the top selected mutations that, like the mutation that was first detected in the ribose operon, deleted or otherwise made large changes in the E. coli genome. And they're almost certainly all destructive mutations, and yet they're beneficial. They help the organism grow faster. So here is the startling thing that we have learned due to the advance of technology in just the past 10-ish or so years. We already knew from about 1950 or so that of those mutations that affect an organism, about 99% are detrimental. That is, they make the organism sick. They make it less successful. The astonishing thing that is, of even beneficial mutations, the great majority break genes or degrade function. They break things, but they don't make the organism sick, they make it grow faster. And that's good, but again, the broken gene spreads in the population, and that loss of information is frozen in time. The take-home points is that now we know what nobody else could know. Darwin didn't know about molecules or cells. Scientists after him could not determine what the mutations were that were powering evolution. But now we can see that Darwin's mechanism is dominated by what I call poison pill mutations. That is, they are positively selected, helpful, but they are loss of function. And that Darwin's mechanism works chiefly by squandering genetic information for short-term gain. Puppy dogs. Dogs have long been pointed to as examples of the great power of selection. Look at all these breeds that humans have been able to foster in only the past few hundred years or so. There are breeds with long curly hair and short legs and short muzzles and all sorts of stuff. But because of the great ability to sequence DNA these days, scientists, some of them, were interested in studying these different breeds to see what the mutations were that caused these beneficial traits, beneficial in the sense that they got taken into homes, they got raised, and so on. And the long and short of it is they're pretty much all loss of function genes.